Good day, everyone. So my name is Jomari Bakay. I am a third year college student taking up BS Biology, specializing in microbiology. So for today's discussion, I'll be introducing you guys in one of the widely used medium in the field of microbiology. So this medium is very essential in a sense that it is very specific and at the same time differential in action when it comes to microorganisms, specifically the Vibrio species, like the Vibrio cholerae, the causative agent of cholera, and some other Vibrio species like the Vibrio parahemolyticus. Okay, so let's go to our presentation. Okay, so I'll just share my screen. Okay. Okay. So this is our presentation. Okay. So just wait for a minute. It's loading. Oh, what happened? Okay. So our discussion is about the TCBS Agar. So TCBS stands for the Thiosulfate Citrate Bile Salt Sucrose Agar. So as you can see, this is an example of the agar, your TCBS agar, consisting of Vibrio cholerae species. So as you can see, the agar is turned to be color yellow as we observed it. It's because of the presence of these um, microbial colonies present in your medium. Okay. Okay, so what is TCBS Agar? So again, TCBS Agar stands for the Thiosulfate Citrate Bile Salt Sucrose Agar. So the name of this agar suggests us what the composition of these agar is all about. So some of the basic ingredients of these agar are given in its name. So you have thiosulfate, you have citrate, bile salts, sucrose, and the agar itself. So let's more something about these agar. Okay, so the type of the agar. So this type of agar is selective differential medium. So meaning to say, it inhibits the growth of other microorganism. It is very selective in a sense that the only microorganism that can grow here is the Vibrio species. So this agar is highly selective and differential only to Vibrio species. So in a sense, you cannot grow other type of microorganisms using this type of agar. So that's the definition about selective and differential medium. So we use this agar in isolation and cultivation of microorganisms. So in the laboratory, we use agar specifically to grow microbes, to culture them, and to use them, and also to isolate them. We usually transfer this from one agar medium or from one nutrient medium to another. That's we maintain them because we use them in series of tests. Okay, so that's why we have to culture them using various nutrient medium. So in this type of agar, it is highly emphasized that it is used for the cultivation and isolation of your Vibrio species. So, for example, the Vibrio cholerae, which is a causative agent of cholera. So this type of bacteria is a gram-negative bacteria. So just to illustrate, here on the upper left side, this is your Vibrio cholerae species. So as you can see, they are comma-shaped bacteria. So this is a highly causative agent of a disease, the cholera. That is, when you get infected by these disease or this illness because of this bacterial agent, you got to experience gastrointestinal problems like vomiting, stomach ache, and so many others. So we can track them because they commonly attach themselves to the chitin covering of seashells or seafoods like crabs, like any marine organism that has shell or have shells on them. They're highly attracted to the chitin, so they assemble themselves, forming a colony, or forming being attached themselves to the chitin containing compound of such seafoods. Okay, so to give you an idea, side note only, the proponent or the discoverer of these TCBS agar. So TCBS agar was firstly developed by Nakanishi. So we're just using their last name for reference. And as time goes by, because there's a development in the field of science, in the field of biology, microbiology, it was modified by Kobayashi and his colleagues. So that's it. Next, okay. So we move to excuse me, we move to the composition of these type of agar. So what comprises the TCBS agar? So as you can see here on the left side of our presentation, there's a list of the ingredients that is included when you buy a prepared TCBS agar, because you can buy this agar prepared, in a prepared form. So it's your the thing that you will do only after buying this TCBS agar is the preparation method, and also the isolation and the maintenance. Okay, so here in the list we have series of ingredients. So to rank them from, in terms of the number of grams per liter. So in number one, we have the sucrose. It accounts for 20 grams per liter. So as you can see, sucrose is the major composition or major ingredient of these type of agar. It accounts for 20 grams per liter. Okay. So next is the agar. So your solidifying agents account for 15 grams per liter. And it was followed by proteose peptone, sodium citrate, sodium chloride, sodium thiosulfate for 10 grams per liter each. So meaning to say, somehow your agar is kind of salty because you have sodium at the presence of sodium. Okay. So in the fourth spot, we have the bile. That's for about 8 grams per liter of the agar or agar medium. Number 5, we have the yeast extract for accounts for 5 grams per liter, followed by ferric citrate, 1 grams per liter. 7, we have bromothymol blue and thymol blue as an indicator or as indicators, that accounts for 0.04 grams per liter. So that's the least ingredient present in our agar. So these agar, we are maintaining a final pH of 8.6 plus minus 0.2 at 25 degrees Celsius. So in that sense, we can say that our agar is basic. Okay. So let's move to the principle of these TCBS agar. So the name itself suggests the name of these agar suggests the ingredients or the composition it has. So we've discussed a while ago the composition of TCBS agar, and now we'll be talking about some of the present ingredients and their action on how this TCBS agar works as a selective and differential medium. Okay, so in number one, we have the protease, peptone, and yeast extract. So in harmony, they work together to provide the nutritional requirements. So meaning to say, these two ingredients are the source of nutrition of our microorganisms that are growing in our TCBS agar. So it provides some nitrogen compounds, vitamin C complex, and many others that promotes the growth of our microorganisms, specifically the Vibrio species. So in number 2, we have the bile salts and sodium citrate. So it it is an agent that inhibits the growth, or agents that inhibit the growth of gram-positive bacteria and coliforms. So again, this type of agar is selective and differential in a sense that it only allows the growth of gram-negative bacteria, specifically the Vibrio species, and it does not favors the growth of other type of microorganisms, specifically other type of bacteria. So you expect that gram-positive bacteria will never grow on this type of agar. So next, we move to the sodium thiosulfate on number 3. It is the source of sulfur and production of hydrogen sulfide. So this is a good source of sulfur, as it was mentioned a while ago. And the combination of the sulfur that is being produced by this sodium thiosulfate, its combination to the ferric citrate, detects the production of hydrogen sulfide, which is being used also by our microorganism in undergoing metabolic processes. So on the 4th spot, we have sucrose. So sucrose is the fermentable carbohydrate. So we know that fermentation is part of our metabolic processes. So bacteria utilize sucrose as their fuel in producing ATP, or their energy that can be used within their system to undergo certain life processes, like cell signaling, like what else, the transport of nutrients, transport of molecules in and out of the cell. So those are such things that uses ATP in quite sense. Okay. So next we move to the sodium chloride. So sodium chloride provides optimum growth and metabolic processes. So the sodium chloride is also essential in the growth of our microorganism because it provides other nutritional necessities of our microorganism. And lastly, that is the most important, because your agar cannot be an agar without this, or your medium cannot be a solidified medium without this. So the agar. So agar is used for the solidification of your TCBS medium. Okay. Okay, so we move to the preparation of the TCBS agar. So like any other type of agar, we usually prepare this type of agar, like the nutrient agar. We prepare them accordingly to instruction given by the product itself. Because again, as I mentioned a while ago, we can buy a prepared product in TCBS agar, of TCBS agar in different stores or microbiological stores that offers or that sells this type of agar. So first, we have to suspend 89.08 grams of dehydrated medium in 1000 ml or 1 liter of distilled or deionized water. So that's easy. We have to combine solid reagents and liquid reagents in specific amounts. Okay, so or ratio or amounts. So we use 89.08 grams of the TCBS prepared medium, dehydrated medium, and we use 1 liter of distilled or deionized water. Note, it is very essential or a must for us to use distilled or deionized water to avoid contamination. And take note that we have to observe aseptic, we have to perform aseptic technique prior and at the end of our experiment to avoid contamination. And we also need to, use or wear lab gowns, face mask, okay, in a way to, protect our medium. Okay, number two, upon mixing the solid and liquid ingredients, we have to heat it by, heat it to boiling to dissolve the medium completely. So the principle behind this is that we increase um, the kinetic energy inside the medium that we are preparing, and in a way, because of that, there is a collision that is happening. Thus, it gives a fast reaction to dissolve our solid reagents using our liquid reagent, which is the deionized or distilled water. Next step, or the third step, is do not autoclave the prepared or the dissolved medium. So we're not autoclaving it in a sense that because some of the present ingredients or compounds or present in this TCBS agar loses its ability to to be to contribute to the differential and selectiveness of this agar because of too much heat. So in a sense, we do not autoclave it. Okay. So in number four, we have to cool it to around 45 to 50 degrees Celsius. That is to control or to lessen the heatness present in our agar. And number five, we have to mix it well and pour into our sterile Petri plates. So that's the time that we allow our medium to completely set and solidify. Okay, so take note that we must do it in, if you have safety cabinet, that's very good. And we must just follow our aseptic technique in preparation of our agar. Okay, so for instance, you've inoculated our agar with your bacterial species, specifically the Vibrio species. Now we're about to look at the result by interpreting the morphological characteristics of our microorganisms, specifically their colony forming on our TCBS agar. So for instance, you have Vibrio cholerae in your agar. So the characteristic of um, those species is that the characteristic of those species is that they are forming flat yellow colonies, 2 to 3 millimeters in diameter. So the picture a while ago, this one, so this is an example of a Vibrio cholerae in a TCBS agar. So as you can see, it's yellow, but it looks like gold here because there's some reaction with the agar. So the agar itself changes its color when colony forms because of some chemical reaction. So as you can see, individually, these are colonies. Yeah, these are colonies. And if we measure the diameter according to the morphological characterization given, it's about 2 to 3 millimeters. Okay, or yeah, 2 to 3 millimeters in terms of diameter. Next, we have the Vibrio agenolithicus that's characterized by their large yellow colonies. Vibrio pluvialis, Vibrio volnificus, yellow or translucent colonies. So in here, we can infer that different types of Vibrio species can exhibit, different types of morphological characteristics when examining their colonies. And then we have the Vibrio parahemolyticus, another one. Their characteristic is that the colonies are colorless colonies with green center. So even though they are same genera of genera, they exhibiting different types of um, morphological characteristic in terms of the colony, because as you can see here, there are of different species. Okay, cholera, genolyticus, fluvialis, vibrio, vulniticus, or hemolyticus. So we can also culture here Pseudomonas aeromonas, Enterobacteria, and others. But these are less cultured using the TCBS Agar. So it's very particular to Vibrio species. So, okay, so there are some limitations. Okay, so most of the things that I mentioned a while ago are just, advantage of using this type of agar. So another advantage is that it is very useful in isolation of Vibrio cholerae and enteropathogenic Vibrio species. So we can use this agar when we are isolating Vibrio species coming from our gut. Yes, we can use TCBS agar in understanding and studying them. So second one is that it is being used for the control of outbreaks of the crown of thorns sea star, the Acanthaster plant C. Okay, so we can also use this type of agar. So those are some of the advantages that is also included in using TCBS agar. And we have also some limitations or disadvantages. Okay, so number one, further tests must be performed to confirm the Vibrio species as it is due to the variation of available nutrition. Okay, so a while ago, while revisiting the interpretation of the colonies present on our TCBS agar, we saw that they are varying on the characteristics of the colony. Thus, it is advisable that we must perform further tests, biochemical tests, in order for us to, deduce this type of microorganism into specific species level. Okay, so we can say that it is Vibrio cholerae, Vibrio alginolyticus, and so on. Number two, inoculation must be performed heavily because the fermentation of sucrose and acid accumulation might inhibit the growth of those species. Okay, so it is a must, or it is advisable that we must inoculate it um, seriously and heavily, because these type of microorganisms are somehow very very dependent on how we introduce them to our culture medium. So we have to, it because they are reacting differently on the present reagents or present composition ingredients of our agar. So some microorganisms, even though they are in Vibrio species group, they can exhibit low growth of colony. Okay. And lastly, TCBS agar is an unsatisfactory medium for Vibrio species when performing oxidase testing. So when performing oxidase testing, it is not advisable to use this type of agar. So we have to look for another type of agar that can be used in, when we are testing for the oxidase process of these type of microorganism. So this overall presentation is based on this page, the microbe notes. So specifically, we refer to the Thiosulfate, Citrate, Basalt, Sucrose, TCBS Agar, culture media, microbe notes. So that's all for today. I hope you learned a lot in this lesson. Thank you very much.